Hi guys, welcome to FRQ 2015 review today. This is Caroline with Fiveable. Welcome, happy to have you here, and hopefully we can get some of your questions answered today. Okay, so let's jump in. Okay, as always, follow us on all of our social media. And then today, what we're talking about, we're going to go over the latest updates to the AP exam, and then we're going to do the first FRQ from 2015, and then we're actually going to do two FRQs from 2014. And there's a reason behind that, and I'll tell you guys once we get started, but it just has to do with the types of FRQs that you're going to be seeing on your exam. So that brings me to the free responses that we're going to be working on. And the reason why I'm using some of 2014's as well is because a lot of the free responses from years previous are about either, especially with these categories, experimental design and conceptual analysis, are about ecology or evolution. So obviously I don't want to review those with you guys because that's not the content that you need to be familiar with, okay? And then also I have to make sure that I'm looking for free response questions that either fit one of those two models, right? Experimental design or conceptual analysis. So that's why I kind of picked and choose. And again, it's not going to be absolutely perfect, but it's still going to be really good practice. So let me see what these questions are here. And then, isn't open book cheating? That's a good question. So no, it's not, because it's all going to be application based. So I would guess that the questions are going to be pretty challenging, and it's not going to be like anything that you can find on the internet. So you may be able to find like a really simple vocabulary word or look something like that up, but you won't be able to apply it to whatever situation they're giving you without strong biological knowledge. So we don't know exactly what that will look like. But because there's no way that they can monitor whether or not you have materials out in front of you, they're just giving it to you as open book. And that's what a lot of colleges are doing right now too. And the exams are just very challenging and application based. And so they're still really hard, but you're just, you obviously have information in front of you. And then part of it is time management, right? You can't learn something in 25 minutes and write a full couple page free response on it. So it's not cheating. And a lot of colleges are doing it right now too. When will College Board give us the practice test? That's a great question that we don't have a date for yet, but I'm sure Fiveable will send out an email once it is out. And then I would just check the College Board website because they've been pretty good about updating it with COVID announcements. And so, or follow, I think his name is Ted Packer. Yeah, on Twitter, because he tweets out a lot as well. So I don't have the answer for you, but when we do, we will definitely share that. Rachel, do you think we should write or type our responses? That's really a personal preference. I'm a very fast typer, so I would type. But I also know that sometimes people are more insightful when they're writing. So it really depends on you. Do you feel comfortable typing? Do you like the capability to go back and edit? That's something that I like to do. I like to kind of write everything out and then go back and edit. So it's easier for me to do that on a computer. But if you don't feel comfortable with typing very quickly and you like to write things out or maybe even draw things out, then that might be the best bet for you. So it's really a personal choice, unfortunately. Okay, thanks for your questions, you guys. Okay, so obviously now that we know that it's only free response and it's those two specific types of free response, that's what our focus is going to be, which is why we're obviously going over free response today. So I looked at the two potential prompts, right? The things that I just went through with you, and took out the power words. And the power words are like the action words, what you should be doing. And they're usually bolded in the prompt. And we're going to see that in our practice examples. So we have describe, explain, identify, justify, analyze, and predict. So some of these are easier than others. I think describe and identify are both pretty straightforward. So when you're describing, you're providing information on what is happening, right? So you're maybe looking at a chart and describing what is happening. Then to explain that is to describe why something is happening. Okay, so that's kind of the difference between those two. A description is really just looking at something and telling what is happening, and then explaining is describing why it's happening. So just a little distinction there. To identify is just to, maybe we have to identify a variable, right? And that would just be the independent variable is blank, right? And sometimes I have students write on and on because it is what the scientist is changing. You don't need to do that, okay? They're just looking really for like a one-word explanation for those kinds of things. So identify is pretty straightforward. To justify, you need to prove something. Usually you're using evidence. It's kind of similar to a prediction. So when you're predicting something, you're saying, this is what I think is going to happen. And then you're usually justifying your prediction with evidence. Okay, so that's where you need to prove why what you're saying makes sense or why you think that's going to happen. Okay. And then to analyze is to look at the situation and describe what's happening with your own insight. Okay, so for analyzing, you're taking a look and you're saying, okay, here's what's happening, here's why it's happening, and this is what I think might happen next, or something along those lines. And I know it's hard for me to describe the power words without actually using context, but when we go through these practice-free responses, that should make more sense. Okay. Is predict basically free points because you're just making a prediction? That's a really great question, and it depends on how they're grading you. So we're also going to be looking at rubrics, which will make this a little bit easier, but sometimes it is a free point, but you usually have to then justify your prediction. As long as you justify it succinctly, sometimes you can get points for predicting either of two answers. Like you could say it could be red or you could say it could be blue, but as long as you back it up with data, they might still give you your point. So as long as you're doing a solid job of backing yourself up, it could be seen as a free point. All right, so moving on. So the first thing that we're going to do is the first free response from the 2015 AP exam. So let me pull that up for you guys. Obviously, I have it on the screen for you, but that's kind of small. So we can pull up the PDF as well. Okay, you guys know that all of these, every year is up and available for you to look through. 2013 and on are going to be the ones that are more relevant. And then you just want to search for ones that have experimental data or that are showing you a process and a disruption in that process. Okay. And then I'm going to scroll on down to this first question. Okay, so frequently when you're looking at these experimental design questions, that's usually either question 1 or question 2 of the old AP exams are going to be the experimental design, and they're long. They're worth 10 points, okay? And so this is kind of going to be what this will look like for you guys. And you can see that there's data given here as well. So I'll read through this with you guys and then go through kind of how I would answer this question, and we can kind of talk it out together. Okay, so it says many species have circadian rhythms that exhibit an approximately 24-hour cycle. Circadian rhythms are controlled by both genetics and environmental conditions, including light. Researchers investigated the effect of light on mouse behavior by using a running wheel with a motor sensor to record activity on actograms, as shown in Figure 1. So I'm going to pause here because sometimes students get confused and they're like, I don't understand what an actogram is or what a motion sensor on a running wheel is or any of those things. And that's totally fine. You might not even know necessarily what a circadian rhythm is, although maybe you've learned it in class, depends on if you guys covered that or not, but you can still answer this question, especially parts of this question. So a really important strategy that we'll continue working on is just not getting bogged down in the details. I've heard from AP graders that students will write things like, my teacher never taught me this as their free response answer, which 1 is a waste of time, but 2, a lot of times they're going to throw random situations that you've never seen at you, right? And it's your job to just apply. That's what we're saying. It's not going to be something that you've seen before probably, which is why like having open book isn't going to be super, super helpful because you're just analyzing these new situations, okay? So hopefully that makes sense. I always reinforce that to my students because they get so bogged down by weird words or weird things like actograms, and it really doesn't matter. Okay. All right. So we can see here that this guy is inactive during the first 12 hours and then active during the next 12 hours. So they're inactive during the day and then active at night is what this is showing me for all 7 days of this actogram. Okay. All right. So I'm analyzing this data as I go. So I'm reading Figure 1, interpreting, and then moving on to the next part. Okay. For the investigation, adult male mice were individually housed in cages in a soundproof room at 25 degrees Celsius. Any guesses as to what that sentence is telling us? We have adult male mice individually housed in cages, soundproof room, 25 degrees Celsius. Any guesses as to what that information is telling us? Oh, sorry, guys. I didn't realize that the screen is not bigger. You shouldn't have to look at my face that closely. Any guesses? You can write it in the chat. The controls. Yes, Serena. Awesome. Thank you for participating. All of those are the controls. Whenever you see a sentence like that, that's what you should be thinking. And they very well may ask you to identify the controls. Because again, think about it. You can't just look that up in a book. You could look up what a control is, but you're not going to be able to pick out this specific scenario in like a book or something like that. Okay. So it's good to have your brain thinking that way. All right. Each mouse was provided with adequate food, water, bedding material, and a running wheel. So again, that's more controls. The mice were exposed to daily periods of 12 hours of light and 12 hours of dark for 14 days, and their activity was continuously monitored. The activity data was shown below. Okay, so this looks very similar to the data that we saw above. We just have, mainly inactivity during the light and mainly activity during the dark. Okay, so this figure 2 is really representing almost the same thing as figure 1 in this experiment, which we know is L12D12. Okay. After 14 days in L12D12, the mice were placed in continuous darkness, and their activity on the running wheel was recorded as before. The activity data under DD conditions are shown in figure 3. Okay, so now obviously we do not have this same even pattern of 12 and 12. In fact, we're seeing this kind of shift of activity over time. Okay, so obviously what I'm thinking to myself right now is that light is impacting the activity levels of these mice and when they're active, because when we lose that light, they're still following some sort of pattern, right? There's still a pattern here. It doesn't all go haywire, but it's not following the same pattern as before. Okay. All right. Everyone good so far? Any questions just of what I just read through? Okay. It's important to take that much time to read through all of the information. So I'm trying to model what I would do in this situation, and I would do exactly what I just said. So I would read through, I would think about what things mean, I would look at the graphs, and I would compare them, okay? Because that's half the battle. Now when we're answering the questions, it should be a lot easier, okay? All right, so let's take a look, and I'm going to be asking you guys some questions too. Okay, so the first part is about the nervous system. So some have some outside knowledge, some didn't talk about it. That's okay. They've kind of redesigned that. So a lot of classes will talk about it when they talk about cell signaling or in psych. Yes, awesome, Rudy. Great knowledge. So some have talked about it, some haven't. So that's one of those things where it's not as important that we go through it. But we are going to take a look at this part A then briefly. Okay, so the nervous system plays a role in coordinating the observed activity pattern of the mice in response to light-dark stimuli. Describe one role of each of the following anatomical structures in response to light-dark stimuli. So this is something that I would not expect. So I'm reading through this and we can just talk about what the word describe means, but I would not expect you guys to see something like this on your AP exam. Why? Because you can easily look up what a photoreceptor does, what the brain does, and what a motor neuron does, right? So that's not really strong application. But in order to describe this, I'm going to pull up the rubric really quick and tell you what they expect you to have written. Okay. So the other important thing about going through these FRQs is going through the rubrics as well, because they expect some really specific stuff, okay? For instance, so for part A, for the photoreceptor, I know some of you were talking about afferent and efferent neurons. What they expected is you to say that it detects light or dark stimulus and initiates or transmits a signal. So here's where students lose points all the time, and this is why I want to spend time talking with you guys about writing strong FRQs. Most of my students would write something like a photoreceptor detects light, okay? 100% accurate, okay? But it doesn't describe how this is responding to the stimulus. It detects the stimulus, but then what does it do with it, right? So in order to get this point, you would have to say it detects light or it detects dark, and it initiates a signal or it transmits a signal, right? Because that's what a photoreceptor does. It doesn't just say, ooh, there's some light here, right? It then sends that message up to the brain, okay? So hopefully that makes sense when talking about this. So even though describe can seem like a relatively easy situation, and remember we're focusing on these bold words right now, it's really important that you're answering the question fully and talking about how we're responding to that stimuli, right? Not just that we're detecting that stimuli, okay? All right. For the brain, that would be either integrating or processing or coordinating information. So that's a little bit more straightforward, but you can't just say it receives the signal, right? Because the brain, those interneurons are actually going to work to coordinate that information and then stimulate a response, which is where the motor neuron comes in. So that transmits the signal from the brain to an effector. So you could say to an effector, or you could say to the muscle cells, or you could say, any number of things. But you really need to make those full connections, right? So photoreceptor sends signal to brain coordinates response, and then motor neurons get that signal and then send it along to an effector neuron. Okay, hopefully that makes sense. The I is okay, good. You guys are doing great things. Reaches the threshold potential. That would be another really great thing to say about the motor neuron, is you could say it reaches the threshold potential and then sends that signal. So all of those things would be fair game, but it's just important that you're being very specific and rereading the question. I do this all the time, right? Because when I do practice FRQs, I will miss points sometimes just because I'm not reading carefully enough, right? The other thing to know is that the average on these questions is usually only 3 or 4 points out of 10, okay? So you may miss a point here or there. You can still get a 5. You can still get a 4. You can still get a 3, right? So you have to just be as specific as you possibly can, but try not to stress too much about it, okay? All right. Next, so that's more of a specific content question. You may see something like that because you do have to write in a specific way, but you're more going to see things like these next few parts that say, based on an analysis of the data in figure 2, describe the activity pattern of the mice during the light and dark periods of the L12-D12 cycle, okay? So hopefully now you're realizing how much they can really ask you, even if this is open book, because you can't look the answer to that question up, right? You just have to use the data in front of you. And this is really what they want you to be able to do. So this is a perfect example of a question that you might see. Okay, so in order to do this, so we're describing the activity patterns during the L12-D12 cycle. So half the battle is just finding that again. So I'm going to go up. Here we are, L12-D12, figure 2. Okay, and then we just need to describe this pattern. Okay, so I want you to think about it for a second, how you would describe that pattern in words, and then we can talk about what the rubric says. You can even write it down, jot it down for yourself if you want. Okay, so hopefully you guys have thought of some good descriptors. Active during dark and inactive during light, more activity during the dark. Awesome. Both of those are great responses. Hopefully you thought of something similar. Okay. Describing the activity pattern, you could say active during dark and inactive during light, active only during dark, or inactive only during the light. So both of the answers that you guys wrote were correct and would get you that description point. Okay, so that kind of reinforces what we were talking about earlier with those root words. The pattern of activity appears to follow a period of prolonged activity during darkness and inactivity during the light. Good. My one suggestion to you for questions like this, and they were pretty lenient with their responses, but I would steer clear of saying they are more active during darkness, period. You want to also say, and less active during light, okay? Usually for these questions, they like you to talk about both things, okay? So that's something good to think about, which Rudy did a nice job if you read his response about talking about both, as did Angela. With saying dark and light. Okay, so just kind of keep that in mind. Sometimes the rubric will allow you to just say one or the other. I always tell my students, talk about both sides of the coin, talk about both situations, because you don't want to miss out on easy points when you totally know what's going on and you just forget to write it down. Okay, so hopefully these tricks are helpful and that you have good skills to attack these questions. Okay, so that was an easy point. So here's another thing I want to talk to you guys about. Let's say you get this exact question and you have not talked about the nervous system and you know nothing about it, okay? Do not fool your way through part A, okay? You're gonna skip part A because part B is such an easy point. And as I said, the average on these questions is usually like 4 out of 10, okay? So those 3 points from part A for describing each of those 3 things, you could totally miss them, still get a 7 out of 10 and be way above average. So you kind of have to get over, and I know AP Bio students are super perfectionists, right? I can relate, I totally get it, but you have to kind of go out of order. You can write your question out of order as well, right? So don't feel like you have to answer every part in order. If this was me and I knew nothing about the nervous system, I would start with B, work through and get some easier points based on describing and looking at data and analyzing that. And then if you have time in that 25-minute chunk at the end, you can go back and attempt to answer question A when you have more time and less stress, okay? So again, you guys probably know that from taking tests, but you do not need to answer these guys in order, and you really shouldn't be trying to answer something that you don't know anything about when there might be other easier points waiting for you, okay? Okay. Part C, the researchers claim that the genetically controlled circadian rhythm in the mice does not follow a 24-hour cycle. Describe one difference between the daily pattern of activity under L12-D12 conditions and under DD conditions, and use the data to support. Okay, describe one difference. This is a perfect example of where you can't just say, they're more active in L12-D12, or they are active in the dark during L12-D12 period. You have to talk about the DD conditions as well, okay? So I want you to think about a difference between L12-D12 and the DD conditions, and then you need to support it with data, okay? So that means you need to be referencing something from the graph, okay? So that's going to be 2 points right there that you can use just by looking at these two graphs. So I'm going to scroll back up to the graph. So that's what you guys are looking at. But remember, you guys have the PDF as well if you want to scroll back down to the question. Okay, but that's what I'm going to show you. So take a minute or so to kind of come up with your own response before we go over the rubric. And when we're doing this practice, I often find that it's really helpful to actually write or type these things out, just because just thinking about it, you're like, oh yeah, I totally would have said that, when maybe you would have forgotten a part. So I think it's important to kind of allow yourself to write these things out, so then you can really analyze whether or not you understand what's going on, or can successfully write this for points, because it's hard. It really is. They do not make it easy. Okay, so hopefully you had some time to describe a difference and then support it with evidence. Here are some things that you could have put. So for description, they actually have a number of different descriptors that you could have used. And as long as whatever you said is true, they'll still give you a point for it, even if it's not on the rubric. So that kind of freaks students out sometimes. But as long as the teacher knows what you're saying is true, they'll give you the point. So you could have said for a difference that the active period begins earlier, a little earlier in DD conditions, whereas it stays consistent with L12-D12 conditions. You could have said that the active and inactive period, or the active or inactive period, is shorter than 12 hours each day in the DD condition, whereas it is 12 hours each day in the L12-D12 condition. You could have said that the daily circadian rhythm, because that would be the active and inactive period added together, is less than 24 hours a day in DD conditions, whereas it's equal to 24 hours a day in L12-D12 conditions. Or you could say something as simple as, in the DD conditions, the pattern of activity shifts each day, or it's not the same each day, whereas in L12-D12, it is the same each day. So all that stuff, the answers are in the graph for you. You just need to be able to write out exactly what you're seeing. So if we look at some of these responses, yep, so exactly, Kathit, sorry if I'm butchering your name, says that the pattern of activity and inactivity changes each day. Yes, so the only edit that I would make to that is that you want to be very specific about which condition you're talking about, which you probably knew, but your reader needs to know. The pattern of activity and inactivity changes each day in the DD condition versus staying the same in the L12-D12 condition. You need to be that specific in your response. Okay. Good. And so, Rudy, I'm reading your response also, which looks great. Just make sure for light conditions, you're labeling that as L12-D12. Mice were active during the darknet. Okay. And DD continuous darkness was prolonged and mice were partially active. Good. Awesome. So all of those answers were really, really solid, guys. So that's awesome. And hopefully, if you didn't write your response out, you were at least thinking something similar. All of those were great. Only thing you need to make sure of is that you're being very, very specific, overly specific with your situations. This is what I mean for DD. This is what I mean for L12, D12, okay? Thank you, Rudy. I appreciate it. I'm glad. Do you need any quantitative data, Rachel? That's a great question. So now we're getting into supporting, okay? And love your use of the word quantitative. Shows me you're a true scientist. And you actually don't need to give specific numbers in order to support that claim. You can just say shorter than 12 hours. I mean, that's quantitative because you're saying 12 hours. But if you just say the pattern of activity shifts each day, that's not quantitative, and they will accept that. So no, you don't specifically need quantitative here. So then using the data to support the researcher's claim. Okay, so now we need to remind ourselves of what the researcher's claim is because I forget. Researcher's claim. Let's go back and take a peek. Researchers where's the darn claim? Researchers investigated the effect of light on mouse behavior by using a number of running wheels with a motion sensor. Okay, so they're making the claim that we have circadian rhythms controlled by genetics and environment, including light, and that light is going to affect this circadian rhythm. Okay, so that's what we need to support then with these graphs. So things that you could say is, without light, active, inactive periods are determined only by genetically controlled circadian rhythm. Okay, I love this answer. Oh yeah, good. Awesome, thank you. So what you can say here, sorry, I was just reading Kafit's The Researcher's Claim, which is really, really good. And what you can say here is, if you go back and read, so that's, you saw that I just did that. That's what you guys should be doing too. Scientists are saying that light and genetics are controlling this. Well, we took light out of it. So anything that we're seeing is due to genetics, right? And so you can just say that, and that answer is basically given to you. You just need to be smart enough to find it and then put it into writing, okay? So without light, active or inactive periods are determined only by genetics, okay? Or if it were a 24-hour circadian rhythm, the pattern of activity would be the same as the pattern of activity in L12, D12, meaning like if this was light independent, right? If light was not a factor here, then we would see the same results for L12, D12 as the DD group. So you could even make the claim that light does have an impact on circadian rhythms because we see differences in these two scenarios. So that would be another bit of supporting evidence for the researcher's claim. Okay, does that make sense to everyone? How we're using what we're seeing, there's differences here. So then we know that light is a factor. Hopefully that helps. Okay, perfect. You guys are crushing these responses also. You should feel really proud because these are incredibly challenging. All right, so almost done wrapping this up. Okay, so, sorry, let me just really quick answer Rudy's question. Are all FRQs out of 10 points? So that's a really good question. The long FRQs, so your first 25-minute FRQ will be out of 10 points, but then your second FRQ will be out of less. I'm assuming it'll be out of 4 points. Let me just really quick do the math. What is 10 out of 14? 10 divided by 14. So maybe it'll be 5 points, something like that. But it'll be worth about half as much as your first free response. But I would be, the reason I like to focus on the points is because then you can see exactly where you can achieve points and know that you could describe just one part of question A and still get 1 point. It's not like an all or nothing kind of thing. So hopefully that helps answer that question. But your second free response will be worth less. And we'll take a look at one that looks like that when we look at one of the 2014 free responses. Okay, so let's do. How many points out of 14 do we need to get a 3 or above? That's a great question that I don't have the answer to because a 3 is always the average. So they make a bell curve, okay? So the average is in the middle, and however many points the average number of students gets, that's what they'll make a 3, and then they'll expand from that. So if you have above average, you might get a 4, much above average, you get a 5. Yeah, exactly. So it really depends on what other students get, okay? Good question. Glad you asked. Okay, part D, to investigate the claim that exposure to light overrides the genetically controlled circadian rhythm, the researchers plan to repeat the experiment with mutant mice lacking a gene that controls circadian rhythm. Okay, predict the observed activity pattern of the mutant mice under L12, D12 conditions and under DD conditions that would support the claim that light overrides the genetically controlled circadian rhythm. So we're removing the genetic factor and just focusing on the light factor. So what do you think is going to happen? So this is not necessarily a free point, right? Sometimes we say predictions are free points. This is pretty tricky. So I want you guys to think about it. Okay. Take about a minute and then we'll talk. So sorry, I know I'm interrupting your train of thought, but as you're thinking, you need to describe what you think would happen to the mice, the mutant mouse under the L12, D12 conditions and under the DD conditions. So it's 1 point for each. Okay. So make sure you're talking about both factors. It goes back to that point of why we need to do that. Rudy, you are an MVP. And Lee, great question. It absolutely would. So hopefully you have an opportunity to read through Rudy's answer there or come up with something similar on your own. So Lee, you're exactly right as well. If we only have light to control circadian rhythm now and not genetics, we should still see the same results for the L12, D12 mouse as the L12, D12 non-mutant mouse. Okay, so make sure you're being specific with that language again. I'm going to talk about that a million times over. Okay, but we should see the exact same results. Now, you have more freedom when you're talking about what might happen then in the DD conditions, and Rudy does a really nice job of describing this, and I'm sure you guys wrote something similar, but you have a lot of freedom. You could say that their activity would be random. You could say that they would always be active, that they would always be inactive. That's kind of where this prediction gives you a quote unquote free point. But what you don't want to say is that there's a pattern to their activity, because now we have no light and we have no genetic tie. So we no longer have anything controlling circadian rhythm. And that's what your readers want to make sure you understand, right? You have no genetics, you have no light. So therefore you have no circadian rhythm, okay? Based on the claim that those are the two things that control circadian rhythm. Cool? Awesome. Great job. So those are ways in which we can look at brand new scenarios, right? Like I didn't know about this experiment or that something like this experiment would exist with circadian rhythms and mice, but we can use our good logic and our experimental design practice in order to go through and answer these questions based on the data given. And this is exactly what you should expect from your first question on the AP exam. Okay. We're not going to do part E because part E is all about evolution. You can look at it on your own time, but I'm going to move ahead because we actually only have 20 minutes left. I'm actually going to skip over. I want to get back to this because I definitely want to talk about standard error of the mean. And this question, so this is the FRQ from 2014, the first one, has you doing a lot of identifying and also describing independent and dependent variables. So that's also something that you guys should be very, very familiar with. What's an independent variable? What's a dependent variable? What's a control? Why does a control exist? Right? All of those things you should be practicing with. Okay. So we'll come back to this question if we have time, but this is another experimental design question. And I want to focus on that last question type, which is where they give you a scenario and then tell you what happens if something changes. Okay. So this is question 8 from the 2014 free response. This is only a 3-point question. So yours might have, 1 or 2 more points. I'm not positive how they're going to grade that. But this is only a 3-point question. You can see you're providing, you're describing, and you're describing. Okay, so the question says a research team has genetically engineered, sorry guys, I just realized that I didn't make this big again. Here we go. A research team has genetically engineered a strain of fruit flies to eliminate errors during DNA replication. The team claims that this will eliminate genetic variation in the engineered flies. A second research team claims that eliminating errors during DNA replication will not entirely eliminate genetic variation in the engineered flies. Okay, so we have a regular process. We're disrupting it by eliminating any errors during DNA replication, okay? And then we have 2 opposing viewpoints that we need to analyze, okay? So this is a critical analysis question. This would be more like your second question where you're going to be analyzing some sort of scenario, okay? So the first part A is to provide one piece of evidence that would indicate new genetic variation has occurred in the engineered flies. So the engineered flies do not have DNA errors, okay? But if we're looking at these flies, what's one way that we could tell that they still have new genetic variation? Sky's kind of the limit here. So I want you guys to think about it for a minute and then we'll talk. I see some good stuff up there. Yes, good job, guys. Everyone gets a point. Awesome. You guys are really, really thinking here. Okay, perfect. So everything that you guys said is spot on. So here's the whole list of things that you could have written about. New phenotypes or new genotypes, which a lot of you wrote about in the chat, which is awesome, right? Because if we have a new phenotype, we know then we have new genetic variation. We have a new genotype. Okay, so you could have said either of those, different DNA sequences, which I saw some of you writing about, or different codons. You could use that language as well. Chromosomal differences, different mRNA sequences, or proteins with different amino acids, right? So all of those things going from really, really teeny tiny to larger visual things would all prove to us that we have new genes in that population, right? And that's all that they want you thinking about. So they're not coming out and saying, how could you tell if there's new DNA, right? They make it a little bit more confusing, but then they give you this wide range of answer choices, all of which prove that we have new DNA present. Okay, so you guys did a really solid job answering that. Awesome. Part B, describe one mechanism that could lead to genetic variation in the engineered strain of flies. Okay, so we have no DNA errors. So you can't say, unfortunately, well, random mutation. Ooh, let me see, Tim, if they have that on there. I don't think they do have that on there because random mutation, I would just assume random mutation is basically this error during DNA replication, because that's really how we get random mutations, is because of an error during DNA replication. So I would steer clear of that and think about maybe some other way besides something that involves DNA replication that would lead to genetic variation. Okay, so think about it for a second. I'm seeing some good things. There's also a bajillion things that you could write about here, so you can really be creative. Cool, guys. All good responses. Yes, absolutely. Okay, so now you can see this isn't something that you can just look up, but it's something that you're applying based on your knowledge, and you guys have really great responses. So here's the whole list. So one big one is sexual reproduction, right? Which a lot of you guys kind of referred to, but that's something to remember. When you have sexual reproduction, you're combining two gene pools. You're going to see different things, different variation, right? That's a huge, huge thing that we talk about with that. Meiosis. Okay, so new combinations, crossing over. A lot of you wrote about independent assortment, random fertilization. Remember, those are the three main ways that we see genetic variation through sexual reproduction. Immigration, which I saw someone write about, or you could explain gene flow, new alleles, new genes. They even have viral infection inserting DNA into the genome, which is crazy, but yes, it happens, and you could write about it. Non-disjunction, which I saw someone write. Chromosomal rearrangements, which I saw someone write. And then radiations, chemicals, or mutagens. So I'm pretty sure that you guys wrote almost every single one of these in the chat, which is awesome. So all of those things are things that you know, but sometimes it's hard to take your knowledge and apply it to this random new situation. So just trust yourself, because all of these things you've talked about, and a lot of you guys have written this on the right-hand side here in the chat, and they're all absolutely true. Okay, so all of those things would lead to genetic variation. Okay, so then our third point here comes from describing how genetic variation in a population contributes to the process of evolution. So because we're not talking about evolution, genetic variation is the key to evolution, right? Because it gives us new phenotypes and genotypes for natural selection. But because that's not on your exam, we'll skip that and go back and focus on, with our 11 minutes left, we'll go back and focus on that first free response question. I'm just going to pick a few parts for us to work on, so that you guys have good exposure. But hopefully this shows you how we can see. So basically, what's this question about? It's about DNA replication, right? So that's the process they're explaining. The disruption to that process is removing any error, and then they want you to predict what the results would be, okay? Yeah, absolutely. Good answer over there in the chat. Okay, perfect. All right. So, well, you need to know about body systems for the exam, Angela. That's a great question. I would say no. It may show up as an application question for cell signaling because things like endocrine, the endocrine system would be long distance hormone signaling. The nervous system is synaptic signaling. The immune system is direct contact signaling. So I'll probably do a review on cell signaling. I'll definitely be doing a cram on it where I'll highlight those examples, but you should still be able to answer the question without a specific knowledge of those body systems, okay? So you won't have to know every single nuance of those systems. You'll just need to know basically those general things. What about the kidney for homeostasis? No, you would not need to know about the kidney for homeostasis. I don't believe. I can't imagine they would put something like that on there. Yeah. Good question. Okay, so let's take a look back then at this first free response from 2014. So it's talking about trichomes. So it says trichomes are hair-like outgrowths of the epidermis of plants that are thought to provide protection against being eaten by herbivores. In a certain plant species, stem trichome density is genetically determined. Okay, so you kind of have to take a step back and realize that you don't know what a trichome is. You might forget what epidermis means, but you can still answer this question because we know they're just little projections that are protecting them from being eaten by other things, okay? And that it's determined by genetics. So that's a good example of just like taking all the mumbo jumbo out and just focusing on what the question is saying. To investigate variation in stem trichome density within the plant species, a student counted the number of trichomes on the stems of 6 plants in each of 3 different populations. The student used the data to calculate the mean trichome density for each population. The results are provided in the table below. So we see the 6 plants from each population and the mean. Okay. So the first part is on the axes provided, create a graph. We're going to skip this part, and I'm going to actually skip about talking about standard error of the mean for now, because that's something that I'll talk about closer to the AP exam. And because we only have 9 minutes left, I really want to get through parts B and C. And obviously you guys aren't going to be, or I'm 97% sure you're not going to be creating a graph here, because some of these are just going to be typed out responses and they're not going to make you make a graph on the computer. So it sounds more like you're going to be analyzing a graph, not creating one, okay? All right, so let's take a look at parts B and C then. Okay, part B, based on the sample means and standard errors of the mean, identify the 2 populations that are most likely to have statistically significant differences in the mean stem trichome densities and justify your response. Okay, so, let's talk about this really briefly. So, standard error of the mean is a calculation that tells us basically how much error is there in this number. Is this mean really that good at representing these 6 plants? Okay, how good is our mean doing at being a mean, right? It's kind of like a progress report. If this number is really high, it means there's a lot of error in this number. This number does not adequately represent the population that it's supposed to represent. Okay, think about like a leader. Is that leader representative of the population? If not, we have a lot of error. Okay, that's just a different way to think about it. So what we usually say is that we take the mean and we do plus or minus 2 standard errors of the mean in order to get a range of where we are 95% sure the actual mean for this sample exists. Now that's really hard to think about, but let me give you an example. For population 1, the mean is 9. The standard error is 1. So we're doing plus or minus 2 standard errors of the mean. So I'm going to plus or minus 2. Okay, so that means for population 1, we are 95% sure that the actual mean for this population is between 7, 9 minus 2, and 11, 9 plus 2. Okay, so my range of means for this population is 7 to 11. Okay, I'm going to type that out. Population 1, 7 to 11. Okay, that's my range. And the nice thing is, if you were doing this question on the exam, you would have graphed this out already and it would be easier for you to visualize. But standard error very well may play a role in this exam. So I want to make sure you guys understand it. Okay, so next our mean is 11. Okay, we're going to do the same thing, plus or minus 2 standard errors. So I'm going to subtract 2. So for population 2, subtract 2 would give me 9, and then add 2 would give me 13. So our range of means is somewhere between 9 and 13. We're 95% sure it's in that range. Then lastly, population 3. Population 3, we're going to add and subtract 2 from 14, so we'll get a range of 12 to 16. Somewhere in there is the actual number of trichomes per centimeter squared, okay? I know this is a really weird concept, but this is how statistical analysis works, okay? We can't just look at the mean and say that represents that population. We actually need to do some error analysis there, okay? So now we need to identify the two populations that are most likely to have statistically significant differences in the mean, okay? So in looking at these populations, here's what I notice. Population 1 and 2, their means may overlap, right? Because we go from 7 to 11, and then we go from 9 to 13. So there's that overlap from 9 to 11, from the low end of population 2 and the high end of population 1. If you graphed this out, it would be easier to see because you would actually see that overlap. But population 1 and 2 actually might have the same mean because maybe they both have a mean of 10, right? That number fits in both of those ranges. The same is true with populations 2 and 3. They could both have a mean of 12 or 13 because their ranges overlap between 12 and 13, right? The end of population 2 and the beginning of population 3. So the only ones whose ranges have no overlap is population 1 and 3. So that's your answer then. Identifying the two populations, it's population 1 and population 3, because within 95% certainty, those two ranges of means do not overlap, okay? What numbers you needed to add? Yes, Urena. So you're always going to add, so you can see it here. 2 standard errors of the mean is standard. You're always going to do plus or minus 2 standard errors of the mean. And usually they'll tell you that too. But we have within the 95% confidence, we're going to add 2 standard errors of the mean. So where do I get the standard error of the mean? This right-hand column. And it just so happens that the standard error is the same for all 3 populations. That is not always true. Okay, but here the standard error is all the same. It's all 1. So I need to plus or minus 2 of those. So I'm taking 2 and I'm multiplying it by 1. So I'm plus or minusing 2. So I did that for all 3. I added and I subtracted 2 from each of these means. And that's how I got my range. Okay. Awesome. You guys are great. Thank you kindly. Okay, so then justifying your response would just be saying that their range of means has no overlap. Okay, so that's how you justify using data. So let me just actually see what they specifically want you to say, just in case that's not perfect. So, yep, so you could just say that the sample mean plus or minus 2 standard errors is less than or for population 1 is less than the sample mean plus or minus 2 standard errors for population 3, or that their error bars don't overlap, or that their percent confidence intervals don't overlap. Okay, so any of that language would be your justification. So that's where you're really using the data to justify what you identified. And for identification, all you need to say is population 1 and population 3. Okay? All right, you guys rock. So then let's take a look at part C, and that's how we'll end today. Okay, so describe the independent, dependent variables and control treatment for the experiment. Then we're going to identify an appropriate duration to ensure that natural selection is measured. We're not going to do that, and predict the experimental results that would support the hypothesis. We're not going to do that either. So the only thing I want you guys, because that's all evolution stuff, the only thing I want you guys to do, and you can definitely do this in the 2 minutes remaining, is determine the independent, dependent, and control variables. Okay, so think about that for a second, and then we'll review. Okay, so our question here is that higher trichome density, we're testing whether or not higher trichome density in plants is selected for in the presence of herbivores, right? Because these trichomes are supposed to protect them against herbivores. So our independent variable would be the presence of herbivores, right? That's what we're changing. So we'll probably have a group that don't have herbivores present and a group that do. And we want to see if the group that does has an increase in their trichomes over time. Okay, so that's what you needed to say for independent. Dependent then is super easy. Remember, dependent is always what we measure. So that's going to be the trichome density. So the independent is the presence of herbivores. The dependent is the trichome density. And then the control, you have a lot of freedom here. Okay, there are a lot of things that you could put. You could just say the absence of herbivores. That's a really good control treatment, right? Because we're adding it and then we don't have it. That would be our control. Or then you could also go, well, I guess it says control treatment. So that's really what you would need to say then, because it's not really other controls, but it's the actual treatment you're using, which would just be no herbivores, because that's normal. So we have no herbivores, and then our independent variable is adding in those herbivores, and then we're measuring the trichome density. Okay. Close. Remember, independent variable, Ved, is what we change. I'm just reading your response. You said independent variable is trichome density. We're not changing that. We're measuring that. So that's your dependent variable. Oh, you said, sorry, I didn't mean that. Perfect. Okay, cool. So how do you recommend really understanding the information from lab experiments, since it can sometimes get confusing and I forget what I just read? Angela, that is a very normal experience. So you should feel very normal about that. I would say a couple different things. It's much more important that you understand the concept behind the lab than the lab itself. I would say, for one, tune into our 8 p.m. sessions that are lab reviews, because Jessica does a really nice job of simplifying. And then two, I would Google explanation of AP Biology diffusion and osmosis lab, right? Because AP has structured labs, there will be other resources out there. There might even be another student's lab report out there that you can read through. That's what I would suggest. Or I think Khan Academy or Bozeman might have little like 15-minute videos on these AP labs, because again, they are standardized. So it might be important to just look at them explaining it, so that you can make a little bit more sense of it. So I would just say, start finding some resources. Is using 5 resources enough to get a 4 on the AP Bio exam, or do you recommend any outside resources? That's a really hard question to answer, because it depends on how much studying you're doing on your own. If you're using the study guides, using your textbooks, using your class notes, then you could definitely get a 4, but you have to really be using all those resources. Otherwise, I would say you need to start supplementing with videos. I love Bozeman. You could use Khan Academy, but I would say use our study guides as your jumping off point, and then find resources to fill in any gaps that you might have. Okay, Rudy, what would happen if the average score on the AP test is relatively high? It would just mean that it's a lot harder then, or you need to get a lot more points to get a 5. Yeah, so it would just shift that bell curve up. So hopefully they're making it hard enough where that's not the case, but that's what would happen. I guess if it was really, really high, then just more people would get 4s or 5s. Okay, a control group. So a control group is the group that you are leaving alone, that you are comparing your independent variable group to. So in this case, our independent variable, so I think it's most important to find that first, is adding herbivores. That's what we're doing as scientists in order to see if it has an effect. The control group, we can't just do that. We need to compare it to something. So what are we going to compare it to? We're going to compare it to a group that does not have herbivores present, because that will give us the data that we need in order to look at both equally. Okay, we can't just look at data that we get from the herbivore group. We need to compare it. So the control group is what you're comparing it to. So hopefully that answers your question, Angela. The answers for C, independent variable, was the presence of herbivores. The dependent variable was trichome density, and then the control treatment was no herbivores present. Okay, one more question. Briefly repeat how to justify in B. So you're justifying by saying that your intervals, your 95% confidence intervals, or your standard error bars do not overlap. Okay, we can't have any overlap with those. And Lee, if you're still confused about that, we should definitely spend some time going over that. So maybe join me in a cram session sometime and I can go through that with you. But basically you want to say that the range of possible means does not overlap. Okay, guys, this has been great. Thank you so much for your time. So next week we'll be doing more FRQ practice. Rudy thinks they're fun, so that's awesome. I think they're kind of fun too. Once you get the hang of it, it really starts to click. But it's really important that you spend time on these, because obviously it's going to be your whole AP exam. So you want to make sure that you spend enough time to really understand them fully. Okay, so next week, same time, same stuff, different year, different questions to focus on. But I really appreciate you guys being here and participating so much. It's a lot of fun for me too. So thank you, thank you. And I look forward to seeing you guys again. Well, so thank you guys. Enjoy the rest of your day. Stay safe. And hopefully I see you again. Bye.